Gustavo CAETANO-ANOLLES, qui est un des chercheurs les plus intéressants actuellement dans la mise en perspective de la classification des virus. Donc, et en particulier, il vient de finir un travail publié en Science, comment ça s'appelle, Neuroscience, Advanced Science Advance. It's new, c'est Science Advance, qu'il faut lire quand on s'intéresse au virus et au virus géant en particulier, parce que c'est un travail monstrueux qui permet de faire remonter l'origine des virus à l'origine des cellules, ce qui est un débat très très important actuellement. Cette approche scientifique par l'analyse bioinformatique des données disponibles est une chose qui est très spectaculaire. Il y a plusieurs travaux qui sont très très rafraîchissants dans la manière de concevoir les choses. Thank you for coming here and thank you for your wonderful work.
Thank you. Thank you, Didier, for the invitation. It's very kind of you to have me here and of course talk about two of my favorite subjects, which is evolution on one side and structure on the other. These are the two core things that I love, and that's the reason why I'm doing the kind of things that I'm doing. So I don't know if you hear me well, or should I use the microphone in the back? Can you hear me? Yes? So if not, just let me know and I'll use the microphone. So basically, I wanted to open my talk with this beautiful sculpture from German. Okay? And this is a person that uses glass to basically illustrate the diversity of microbes in general. So it's a wonderful concept and an interesting aspect, to open the talk of viral diversity, because ultimately I'll be speaking about evolution and origin of viruses, which are two difficult subjects, difficult to tackle for many reasons, which I'll try to focus on some of them. The reality is that there is incredible diversity of viral forms and viral genetic material and strategies that are being used for extending or that aspect of genetics in viruses. And on one side, beautiful and diverse morphologies are exemplified here by the various forms that you would find in archaea, and on the other side, incredible diverse genetic strategies. The Baltimore classification groups them basically in 7 groups according to the use of nucleic acid material and also the use of a strategy to ultimately produce messenger RNA. And the interesting thing is that there's only one out of these in cells. So it means that viruses are incredibly diverse, compared to cells. They're just opening an incredible field that we have to explain. And this is the difficult part, because when you're looking at classification point of view, then you have the international community kind of settled in the 7 orders, but yet places 78 other families of the very few that are here in an unclassified group, meaning that we don't know very much. We know very little. It's patchy. A patchwork of information is what we have today in terms of relationships at the higher levels of classification of viruses. So this is kind of the situation today. We have to find ways to unify the group of viruses and then think of ways to relate that group of viruses to the domains of life as we know them, the cellular domains of life, or superkingdoms, whatever term you would want to use for that. Our approach has been the evolutionary approach. And this is a very specific approach, means it's idiographic, meaning that it's historical. So it means that we have to apply algorithmic implementations that will travel back in time and will be retradictive, will the extracted information that is not present today. And this is a big difference, because science in general is what is called nomothetic. It's predictive and universal. We are always finding universal principles, isn't it? Universal principles, and then applying statistics to say, well, this universal principle is of generic importance enough to merit perhaps even a law if we like to go that route. So, but this is historical. This means it's subjected to chance and necessity. And this is a big issue, big difference. And the pitch I'm going to do today will be to tell you that three-dimensional structure of molecules in general, but I will focus on proteins in particular, is the way to go and perform a back-in-time exploration. You know, and that is kind of the message that I have for you basically today. And of course, we want to meet a time traveler. You know, that is the goal. And our time travelers are algorithms. You know, they're exploratory tools that look at the sequences that are real things that we retrieve from genomics, and the structures, which are the real things that we retrieve from structural biology. And we look at those and we apply principles of thought to extract useful information. And then we start asking questions, and I'm going to ask questions today. Where do viruses come from? What is the role of viruses in cellular evolution? Is there a truly universal tree of life? How are viral groups related to each other? Is there a preferential direction of gene transfer? You know, is it going viruses to cells, or is it going cells to viruses? Can we go back billions of years in time, back in time? Can we do that, really? So these are important questions, especially because there are big challenges. First of all, traces have faded. Methodologies have a multitude of limitations, and we're still struggling to solve some of them, and I'll show you some in a minute. It is easy to create theories than to put them to the test. So there are many papers that will put forth hypotheses about viral origins, but is there a way that we can garnish data and use it to put them to the test? And ultimately, if we can these hypotheses be supported by large-scale data? Because the more data we have in the time of data wealth as we are today, then the better we can do. So one aspect that is fundamental for viruses, but it may be also fundamental for deep phylogeny in general. So going really back deep in time and claiming that these big groups are related to each other implies a jump of faith when you are using, for instance, sequences. And molecular sequences has been the entities or the elements that we've used over and over again to make the vast majority of statements, of evolutionary statements today. And I'm going to contest that. I'm going to say, wait a minute, perhaps molecular sequences are not the best way to go. They are wonderful as elements to relate organisms that are closely related to each other, or relatively closely related to each other, but can we really go back in time? And I want to contest this by the fact that the genes are not the only evolutionary units. There is some other levels of abstraction in structure in general, and one of them is the protein domain, and I'll use them, because we use them to extract information. And protein domains are basically building blocks, you know, that are incredibly conserved. They are recurrent in biology, and you can identify them very well, not only in three-dimensional structure, but you can do a good job identifying them from an algorithmic point of view, computationally. And they are compactly folded, they hold the functions, you know, and they are evolutionary units. So how can we reconcile the gene as an evolutionary unit and the domain as an evolutionary unit? So this is an issue, and here is the problem that I'm going to address in a minute. So we have an alignment, and this is the way how we do, isn't it? We find homology, means that we are finding relatedness from an evolutionary point of view, and we claim that a position in an amino acid sequence is homologous to many others. And we do that with incredible tools these days, and then usually when we build an alignment, that already in itself is a tree statement, is an evolutionary statement. So as soon as I do that, then I have a problem. If this protein is a multi-domain protein, and it has two domains, domain A and domain B, and we can assign those domains to some of these sequences, we trust that that assignment applies to all the sequences in an alignment. But what if some sequences do not have that architecture? What if there is an inversion? And this is very common in biology. And we have domain B appearing, more towards the 5' end of the sequence at the nucleic acid level, and domain A, differently located. So when we attempt, in these circumstances, to make an alignment, then we either disregard domain B in the alignment, or we disregard domain A. But we ultimately disregard a portion of the information in the alignment because it needs to be incorrect as soon as we have this heterogeneity in domain makeup. So this is a problem. And I'm going to illustrate this problem with this very recent paper that had a lot of visibility. It's the paper from Spang et al. That was published in Nature very recently. They uncovered this RPO microbe from a metagenomic sample in the Lockyer Castle hydrothermal vent of the North Sea, and they named this the Loci archaea as a new group that amazingly had a number of features that were very close to eukaryotes. So this was used to build a concatenated alignment. This is the merit of the entire paper. The concatenated alignment of a number of highly conserved genes that they painstakingly selected, including RNA polymerases, aggregation factors, aminosyldeonase synthetases, a whole bunch of things that are highly conserved and universal. And then they built a phylogenese, and many phylogenies related to their data set, and they find that the three domains of life should be redefined into two groups, basically, because Loci-archaeum arose in the middle of archaea, but very close to eukaryotes. And eukaryotes were placed within the archaeal group. So this is a favored scenario of evolution that differs very much of the three-domain concept put forth by Carl Rose and many others during the past decades. And it has to be explained in some way. So it's of relevance. And so we looked very carefully at the alignment. We went back and did some bioinformatic analysis of this. And it turns out that when, instead of using maximum likelihood, we use a distance approach to visualize this diversity, using the same data. It's the same alignment. We have not built anything odd out of that. We use that, but we used a different methodology. And the good thing about this methodology is that it produces a network instead of a tree, showing that if there's conflict in the data, that conflict is going to be revealed in the network. And what we find is that we have a three-domain scenario. So the gray and the black are archaea and eukarya. And then the large sampling of archaeas that we use here, including the Loci-archaeum group, they are all placed together in a group. And as you can see, if you go and zoom into the core, there's a lot of convergence or horizontal elements basically going on. As you can see, that core implies that there's a number of things that have to be explained. So, but we did the same thing with domains. So now we split everything into the tiny pieces I was telling you about, and then built a tree from its occurrence using the same methodologies. And again, we have a situation of a three-domain organization of cellular life. So then we went carefully into annotating the alignment and see whether the domains that were in the alignment were consistent throughout the alignment. And to our amazement, we find wherever those little dots in the different archaeal clusters of homologous genes, which are called cogs. These are the R-cogs. And for each R-cog that was used in the concatenated alignment, many of them had inversions or mismatches in the domains. So we are in the problem that I just presented to you, where you have a domain that has been flipped, and you are basically miscataloguing that information, you know, and retrieving it in a way, in a faulty manner by doing this. So this exercise results in that very low level of alignment robustness, where there's a large number of gaps. Obviously, you have to have gaps because you have that kind of problems in a genetic alignment. So, so here, the entire issue I'm bringing you here is the issue of how to deal with structure and how to get that information coupled to the information in sequence when thinking historically, when thinking going back in time. How can we consider that information and extract information that is valuable? Now you may say, well, wait a minute, perhaps these few inversions that were in the alignment of Spang et al. Are a rarity, you know, and this is not something that occurs in nature. So in order to dispel this, we've been studying domain rearrangements from a very global perspective by mining the information in proteomes and using bioinformatic approaches to determine how domains rearrange and trying to build history from that. And this is what you get. You get an entangled network of different domains that are connected by their presence in a protein. And as you can see, it's massive. It's a massive. So we have thousands of possible combinations that are all materializing in the products of our genomes. And they are expressed in that graph. And I'm going to now present you a historical view of this. So at the beginning is the origin of proteins, and when you see the number 1 is the present. And you're going to see how this web unfolds in time, and then you're going to understand what is the methodology that I use later on as I explain this. And so as you can see, these are the accretion process. We are finding new and new domains, that little letters that appear as individual domains that are being uncovered in evolution as time progresses. And every time you see a line appearing, that implies the existence of a combination, you know, a particular arrangement of that, of those two domains. And as you can see, this is becoming dense and dense, and roughly about 0.6 of the scale that goes from 0 to 1, we have a big bang, an explosion. This is probably the rise of metazoa, the rise of recombination and evolution that allows for all this to happen massively all the way till the present, where you have incredible connectivity of individual domains in proteins. So if you don't take into consideration this evolutionary units that are doing all this incredible feat in biology into the analysis of sequences, we are at risk. We are at risk that we may not be extracting all the information that is valuable, and we could be producing biased views. So, one thing that I just want to add, this is very recent work in my lab, is that in music and in language, Menseroth is a German scientist in the 1920s who brought this law, where he found that the larger the systems, the smaller its components. Basically, that was the idea. And this was manifested in the way how we speak, you know, so the information comes in a very compact way whenever we have big constructs of language. And it turns out that with music that occurs too, and it was not until Gabriel Altman in the 50s basically did the math, you know, to understand exactly what was going on, and this is what is kind of recognized as a law, you know, and it's related to information in systems in general, but it's language after all is biological. So we've taken this to the domain combination level, and we find that we have wonderful fitting of a straight line in a log plot of domain length versus number of domains, meaning that the larger the domains, the number decreases. You know, so it decreases. So meaning that larger system, you have to economize and you have to produce smaller entities. So this is universal. We tried 60 genomes doing all these analyses one by one, and we find that there is a correlation, a good correlation for everything. And this is telling us that if there is a law of this kind, this should be incorporated in our models of evolution. We have to incorporate this change in the way how we build a phylogeny. But if we're doing that, we have to elaborate that model in particular, and that has not been done. So, so this is a problem, and it's a problem because we are still struggling to understand macromolecular complexity. It requires holding that understanding. It requires mapping the sequences to the structures, then mapping the structures to the functions, and mapping the functions to fitness ultimately, because that's what selects what we have. And there are many things that we can do. We can think of the full structure, the hierarchical abstraction of the structure, and we will be on the top there. And that gives us insights about evolutionary constraints. But if we are interested in ligands and how they, how the groups that hold the ligands juxtaposed to each other, then we would be interested in functional size. So as you can see, there is an incredible diversity. We could focus in any of these aspects, from interaction interfaces to mechanisms to stability function interface, structural redundancy. So I'm going to concentrate in those two on the top, you know, and left-hand side of the equation, because we feel that structure provides a window into both function and evolution. And the whole idea is that in the protein world, macromolecular world is modular. There are these components that are recurrent, permanently recurrent. They exist already at the supersecondary structure level. Like for instance, we may have alpha-alpha elements or beta-beta hairpins that exist and make up a whole bunch of structures in biology. And they are recurrent. They're permanently there, you know, stabilized probably by van der Waals locks, you know, that make them really compact. And they're permanently revisited in many, many structures. And here is just an example of them. But with RNA, we have the same thing. We have helical elements that are repeated over and over again, and these are modular forms that make up a structure like the ribosome or a complex structure like RNA's pRNA. So they're not only recurrent, but they carry deep evolutionary signal because they're highly conserved. If they're conserved, they have to be canalized, following Wellington's idea. They have to be conserved at that level. And the interesting realization is that as you're traveling in complexity or hierarchical complexity from the sequence all the way to the molecular complex, it takes more and more time to materialize some of these structures. So here I've used the ATP-A system as a way to illustrate the issue. And as you can see, we have only one of those, you know, in terms of a design, with all its diversity, of course, but the overall design of a shaft and an embedded element in the membrane and a rotary piece, you know, it's an electric motor ultimately manifesting in biology. And that's unique. That's 1 out of 3.8 billion years. But on the other side, we have sequences coming up immediately, you know, in all the world, you know, and with incredible diversity on the other side of the spectrum. So we are going to focus on the fold and the fold superfamily as a way to understand evolution. And we go that route because these structures every 1 million to 100 million years, basically, one of these get unfolded. And that's why this is important. So domains, as I told you, are evolutionary units, but they are also, they can be defined at different levels of abstraction. So if I concentrate on the topology, I will concentrate on the code. If I concentrate more on the sequence and the function, then I will go all the way down to family. So as I travel all the way up, there I'll be reaching for deeper signal. At the same time, if I'm traveling all the way up, I'm losing the detailed definition of function. And this is a problem, because that implies that, that we are, in a way, abstracting function at a level of ontology that's much higher than the one we use, for instance, with gene ontology. So we've done that, and we've used both CAF and SCOP, which are the gold standards of structural classification of proteins, to do our deed. And we use something that is of importance, which is the fact that we found that domain diversity that we can categorize as an occurrence, in proteomes in general, and its reuse in a proteome. So let's say we find an interesting structure, like the p-loop hydrolases, and in humans we may have 400 of those in a genome, encoded in a genome. And so that's a reuse level, of that design, over and over again to perform different functions. And so when we do this, we plot, for instance, for a set of genomes. In this case, we have archaea in red, bacteria in blue, and eukaryotes in gray. You're going to see that there is a very linear relationship with microbes, but then you have a kind of departure coming later on with eukaryotes. But this has been consistent. We've done this analysis many times, and we find that they follow what's called a bandwidth distribution. In plots like this, a bandwidth distribution is a manifestation of information in systems. And that departure, that implies an aspect of criticality that separates microbes from the rest of eukaryotes and gives that departure that you have here. So this is telling us that there is information in the use and reuse of structures. And this is important because this information can only be evolutionary in order to build a pattern like this. So this is an encouraging aspect that in my lab we've pushed. And the reason is that we've been for many years interested in understanding protein diversity, understanding the protein world. So how is it made? What are the structures that populate the protein world? How are they connected to each other? Similar to the question that I'm going to be addressing today with viruses. So, and what it exists today is a topological view. But the problem with a topological view is this, is that you can build a network, and this is a galaxy of folds that have been published 3 or 4 years by a group of years. They're of Lupus in Germany. It's a relationship of structures using algorithms similar to GLASS. And as you can see, yes, there is some unity on the structures, but the vast majority of this galaxy are isolated pieces. You can't put them all together. So the topological view that we have of structure is highly isolated. We can't unify the protein world. So a way to solve this has been proposed by William Taylor. And Taylor has brought this idea that I have there, is of a periodic table. So we can abstract the information of a structure to the maximum and then build a table like that. And we plot the signs and then ask, are there rules of change that will relate one abstraction to another? Unfortunately, those rules have not been forthcoming. So we still have a problem, to unify the world of proteins. So we've used a trick, and the trick is the occurrence and the abundance of structures in proteins. They carry information, as I told you, reuse and reuse, and they can be mined in order to extract useful information from them. So now I'm going to go to the gist of phylogenetic analysis. Ultimately, phylogenetic analysis, going back in time, implies, like many things in information, building matrices. A matrix, a numerical matrix. That's the best way to describe a number of things in a world. And mathematicians use it all the time. And indeed, phylogeneticists do that. They build a matrix. And in the matrix, you may look at a system, let's say an organism, and categorize the organism by looking at some of its parts. The parts could be, I don't know, the number of limbs, or it could be a molecule, a molecular sequence, a structure, a domain, whatever. But it's a part of that system. And this is what we've been doing all the time. We've been building since Hegel, basically trees of systems. Trees of systems. Every taxa in that tree is a system, you know, and we're using parts to look at the system. And here's one from our own group. This is a tree built from proteomes. So at the tips of this tree, which has three group makeup, we have individual proteomes that reflect perhaps the system as a whole. You know, they're still parts. A protein is still part of an organism in itself. And the organism is part of entire collective. So it's difficult to dissect, you know, the modules out of the continuum. But that's what we've been doing. And so that implies defining a species, defining a number of things, you know, how we define a system. And then defining the parts, which are as important. So we can define parts as being, let's say, nucleotide sites in a nucleotide sequence or amino acid sequence, amino acid sites in amino acid sequence. But then we have a problem, because if there is structure, parts of those sites may be interacting with each other. That little arrow that goes over there. And if you have that, you have to account that in order to build the phylogeny that is below to describe the system. You have to account that in the model of evolution. If you don't do that, then you are violating the sacred principle of phylogenetic reconstruction, which is character independence. The independence of whatever you're studying, or whatever part you're studying from each other. Because if you are not modeling that non-independence, then you may be producing an artifact. So this is a big problem. And so one thing that is interesting is that we could also study the evolution of parts instead of the evolution of systems. And you see where I'm going, because if I'm interested in the evolution of proteins, of the protein world, then I would want to build a tree that describes all of the structural diversity, let's say, of proteins. And this tree will have as taxa not the systems, not the organisms, or the proteomes, or whatever, but it's going to have the actual entities that I'm studying, the parts, the domains, let's say. You know, I need to define those as my parts. And the good thing about this is that then you can build trees like this, where I'm basically generating a phylogeny of the high abstraction level of the CAF classification. So now we are seeing how the different structures evolve in time. So this is a unified view of protein evolution. But it has an interesting consequence. And the consequence is that if there is any violation, that violation comes from the features that I'm studying, which are systems. And if I'm using proteomes, then the violation will imply that there should be an interaction between, you know, different proteomes to each other. So I can claim that if I have humans and I have Vibrio fischeri, which is a bacterium that lives in the oceans, perhaps there's very little contact between the two, and I will not be violating the sacred principle of character independence. So I'm placed at a different level all of a sudden. I'm placed at violations that occur at levels above the system level, you know, of my system level. And this is a benefit. So my contention is that that focusing on evolution of part is much more robust than focusing on evolution of systems. And that would be terrible consequences, because historically we've been generating trees of life, isn't it? We want to generate the phylogenies of Haeckel, you know, because those are our immediate interest. So this is just a detail of, and I think this distills the essence of the methodology. So we built a matrix, which is in the middle there in a way of a heap diagram. And in order to build this matrix, we perform a census. So we go genome by genome, extracting all with hidden Markov models, which are these machine methodologies that allow you to extract useful structural information from the sequences. And we performed this census with known structures, meaning that our the accuracy of our methodology is very robust, because these are curated through structural entries that define the hidden Markov model libraries. And the assignments in history, experimental history, has shown that the accuracy ranges from 95 to 98% certainty that we are going to get a right structure given this hidden Markov model. So of course things can go wrong, of course, but that margin is very small. And then so we have a census. It's a census. It's a demography table, as we call them. And that table makes a matrix. That is a matrix there. On one side we have proteomes. On the other side, vertical proteomes. And on the other side we have domain structures. And and we are mapping globally, because these are all the domain structures that are known. So we are not missing anything that does not produce structural biology. And the good thing is that in terms of folds, there are very fewer and fewer new folds coming to be discovered. So meaning that we have basically covered the world of folds very well. And we are not expecting too many new unfoldings. So this could be regarded as a very finite system, global, that describes the evolution of proteins with that tree that you have there. It's a tree of parts. And on the other side, we have a tree of systems. This is a tree of life that you have there that describes archaea, bacteria, and eukarya in this case. The important thing comes on the top. And this is what we call a molecular clock of folds. And how did we build this? Well, it's we took particular structures and tried to relate those structures to evidence in the geological record. Like for instance, in deposits, that are very, very old, 3.8 billion years, 3.4 million years. These are regions on Earth that have unearthed areas that are mountainous areas that contain this very old crust of Earth. And then we look at carbonaceous materials there. No, we don't do it, but the Earth scientists do. And then extract those carbonaceous materials and ask questions. Is it porphyrin materials related to metal components? And we find that indeed porphyrin associates with vanadium, and vanadium is a metal that replaces iron in time, and this is very well known. So we've used that, and we've annotated enzymes that are related to the pathways of porphyrin biosynthesis, calculated an age with that tree, and for that particular case, we can then assign one particular age from a time in billions of years. And when we continue doing this with a handful of structures we've carefully selected, then we find out that we have an incredible correlation between the age of folds, this is biology, the age of biology in the geological record. There is a good time map, there is a clock, it's universal, embedded in that tree. And so that implies that, well, this is not just a figment of our imagination. There's something else going on that we have to consider that is supporting these statements of trees of parts. And of course, you can look at the tree of life, and we've looked here with viruses, and this is the paper we published in 2012, where we selected just small groups of viruses of large to big size, and included giant viruses. And this only group of 56 proteins, and compared them to 981 proteins. We did this same analysis, look now at the tree of parts, and with the network methodology, but we've done others, we see again a 4-group system, where you get the viruses as an homogeneous group, different from archaea, bacteria, and eukarya. And then if you do this, which we have now published, but published now very recently, but at that time, we had that data, and we would use the age of the different parts of each individual protein and treat that as a multidimensional space. So it's a space of time, put it this way, isn't it? As a multidimensional space. And then using principal coordinate analysis, which is a numerical technique to mine, spaces that are complicated that we can visualize. We find these are the most important components of this multidimensional space. And what you can see is that eukarya has its own area, bacteria, and viruses has its own area. And that little star there is a reconstruction of the ancestor, of the last universal common ancestor of cells. And it's between viruses and the rest in that space of time. So it was very interesting for us. We said, well, perhaps we are onto something. Can we really go deep into viral analysis? So we've done 2 data sets, extended data sets. One contains 4,211 genomes, and then the very recent one that was published in Science Advances has 5,080 genomes. We've sampled a whole bunch of biodiversity that was available at the time of sampling, 3,460 proteomes of the second set. That's the one I'm going to be talking today. And we focus on one of those levels of hierarchical complexity, which is the fold superfamily level. It's not high enough as the fold, but it's conserved enough to be considered evolutionarily related to the component parts of those. And we use 2 approaches. A comparative genomic, just look at the numbers and what do they tell us? This is not going retradictive, not going back in time. Then the second one confirms any kind of possible inference that we may have in the system is structural phylogenomics, as you may call it. So, and this is the comparative genomic approach. It provides only tentative information, as comparative genomics should. And it starts with a census, careful census, produces demography tables up there, you will be able to see all the details. But then from there, it looks at subgroups. Let's say we consider the group of archaea together, or the group of Archaeota together. And then look at how the number of fold superfamilies distributes relative to that group to the rest. And then in doing that, estimate how it distributes in the world of those earlier Archaeota, for instance. How the component parts distribute there. And so we've done that analysis, and here we are going to do this analysis with this big data set of 5,000 genomes. There are 1,995 fold superfamilies present in that data set of proteomes. And as you can see, this is a Venn diagram, a 4-way Venn diagram: viruses, bacteria, eukarya, archaea. There are 15 groups that we may call 15 Venn groups. And each Venn group implies a statement of sharing, isn't it? So if I have the Venn group of A and E, implies that it's fold structures that are being shared by archaea and eukarya. And if I have the ABE group, these are the fold structures that are shared by all cells. And the ABEB group shared by all cells and viruses. So one thing that jumps at you immediately is this: the vast majority of fold superfamilies are common. And they are in the groups of the ABE and the group of the ABEB. So this implies deep phylogenetic signal, because one of the fundamental elements in Darwinian thought is that we have a hierarchy, isn't it, of nested elements in evolution. So if there's a core that is very ancient, that core is unifying the entire tree. So you have to have something like this whenever you're exploring deep hierarchies. So that's how we take it. The other thing that is interesting too is the fact that viruses share fold structures with each and every one of the other 3 domains of cellular life. So this is, again, an unanticipated finding, because, we could have had incredible different biases. There are big biases in this Venn diagram. And then this is the other wonderful thing, is that the 4 groups have incredible diversity in terms of the things that are unique to each group. So the archaea have 24 fold structures that are unique to archaea, 283 for eukarya, 154 for bacteria, and look, 66 for viruses, meaning that the diversity of fold structures of viruses is larger than the entire cellular domain of life, the archaea. So this is something really interesting, that needs explanation. So why do we have 3 times more abundance of these virus-specific fold superfamilies? They need to be explained. They probably underrepresented, because it is possible that because we are not sampling the viral world, and we know we are not sampling the viral world. I'm not preaching this here to this group because that's what we've learned. Then there are big chances that horizontal gene transfer would make virus-specific folds non-virus-specific. Let's say a single transfer of one lineage, will make a virus-specific fold become a fold shared by some other group. And so that means that there may be many, many folds of that kind. And here I have some interesting findings. These are the 4 viral lineages that have been defined by the capsid structures. And they are good candidates, actually excellent candidates for classification of viruses, because they are shared by quite a large number of viruses that share these capsid structures. And, but yet, look, out of these 4, this includes the jelly roll, the coronavirus-like structure, and the Hong Kong 97-like structure, the third one, they're all shared with cells, meaning that there are variants of this structure in cellular lineages, and yet we consider them not accurate, bona fide, virus-specific fold structures. So comparative genomics is already warning us that we have something to explain here that is important. So when you do this thing, and there's no surprise here, you're on the envoy doing genomics and doing the bioinformatics, you're going to find this. Viral proteins are enriched with proteins of unknown origin. So when you look at the 5,000, you're going to see that the vast majority of proteins with no detectable homologs when you use sequence analysis. Then you're going to see a fraction that is proteins with cellular homologs, and it's a substantial fraction. But then there's still a fraction of proteins harboring virus-specific fold superfamily that is substantial for each of the 3 big groups. There are paraviruses, the bacterial viruses, and eukaryoviruses. So there's some bias here, but it's a substantial element of it. So there's a large fraction of viral proteins that does not resemble known proteins, meaning that viral diversity is generated intrinsically through viruses. Now you could say, well, they could have been captured from cells by evolve rapidly in viruses, but then we know that some megaviruses, their complements evolve at the same rate as normal genes. So then this may not apply throughout the spectrum. We could think that they were captured from several species that are not yet to be sequenced. But then we know that folds at this level, folds of the families, have not been increasing in our exploration of the protein world. So meaning that where are they going to come from if we're not seeing that? So it's quite likely that viruses create genetic novelty. That's probably the explanation of these results. And if they do, then they misspell this idea that viruses are not living entities. They are definitely they're evolving. Okay, so that's nonsense. So and this provides information that supports this contention. So every fold structure that is shared with the viruses, it's spread in the cellular world more widely than the structures that are not. So this is what this analysis on the top shows. So every time that you have a sharing, which is the bright yellow one, the number of the F value, which is the number of proteomes with a fold superfamily divided by the total number of proteins. This is a fraction of how much that structure is spread in the protein world. It's always higher, if viruses are involved. So this is information that viruses in some way enhance molecular biodiversity when it's described at the structural level. But then we have to be careful, because we know virus-host relationships exist, and they are incredible biases, and this is an analysis we've done of how individual superkingdoms basically interact with the viral world. And our sampling of today indicates that bacteria, basically, and archaea are mostly double-stranded DNA viruses. And that's their makeup, and this is basically a quantitative view of that. And that is completely different from the one in eukarya. In eukarya, we have all the replication strategies, all the diversity in the world, yet bacteria and archaea do not have that. So this needs to be explained. Eukaryotes are infected by all types of viruses. RNA viruses are rare in bacteria and completely absent in archaea, but there's been one publication recently that finds that microbial eukaryotes resemble prokaryotes in their virus distribution, and organism complexity is somehow proportional to the number and variety of viral infections. So that would be, this is in agreement with this idea that viruses generate novelty. So, you know, the more interactions with cells, the more novelty they're generating, and that expresses in their replication strategies, for instance. But this is the wonderful thing. When you look at these three large groups, you know, the eukaryoviruses, the archaeoviruses, and the bacterial viruses, and you look for a structure that unifies them. In this Venn diagram, again, we have at the middle there 68 out of that world that we sampled. A substantial number of things that unify the world of viruses. And this is good news, because that implies that if we have that, we can mine that information for our advantage to go deep in time. So that's exactly what we've done with this thing. This is comparative exercise. You know, so we've used this kind of centrality or core aspect of biology to build then this kind of thing. You know, this is a 7-way Venn diagram, and it's feasible. Not all Venn diagrams can be visualized as this. The 11 Venn diagram can be visualized, but not others. So it's odd. But this Venn diagram shows something very interesting. The 67 full superfamilies that are shared between viral supergroups with more than one viral subgroup can be summarized in 17 out of 120 possible Venn groups. So it's only 17. That means that it's incredible bias. You know, a bias like this has to come from an evolutionary point of view. There's no other way to explain this incredible bias that we have here. And we believe this comes from extreme genome reduction. We've been, viruses have been losing a lot of material. And this, every time there's loss, there is less information in this sharing aspect of things. So we have to mine in some way this Venn diagram in order to extract phylogenetic deep signal. And this is what we've done. We've gone into this idea of Ariadne's thread. You know, Ariadne is a mythology of using a thread to go through the labyrinth and, you know, in a plot to kill the mythological feature, you know, of complexity, in a way, you know. And interestingly, Ariadne threads are also computationally an algorithmic, in a way, implementations, finding basically options that are viable through a sparse area, you know, of complexity. And what we've done is basically look at how many, for instance, possible groups exist of Venn groups that are shared by 4 to 6 groups. So this is what you have. This is a distillation of the findings. And of course, we then count individual full superfamilies that match this distribution. And what we find is that the vast majority of threads are concentrated in the top part there, shared between double-stranded DNA viruses and minus single-stranded RNA and plus single-stranded RNA. That's the core, basically, element, component here that keeps everything through the threads together. And so this is indicating perhaps an early origin of RNA viruses. You know, that we took that element as a possibility that would exist. So can we assign ages to these threads? Because if we do, we could go back in time, and then we could confirm indeed that those threads have historical information. So we have to reconstruct now and go back in time. So this is just a slide to let you know that accretion, which is what we've been studying with the protein world while building those phylogenies, is a universal phenomenon. It occurs at many different levels. It occurs in galaxies, it occurs in stars and planets. This is how we believe our solar system has been formed. And it occurs in proteins because we have to comply with the principle of continuity. Each and complicated element of biology has to unfold gradually in evolution. They can't come all together. And that's an accretion process ultimately. With cities, we have a wonderful studies very recently in the Santa Fe Institute that show that cities basically grow by exploiting super exponential processes. And these super exponential processes are related to what I mentioned earlier of the Benford distribution, remember? So cities, which is a construct of humanity, are growing very much as many aspects of biology do. So, and you can study this thing with the same methodologies. Like for instance, there is an astrocladistics that uses cladistic methodology, phylogenetic reconstruction methodology, to build the history of galaxies. But you can do this with many, many different kind of objects, inanimate. So, so this is the tree of parts. It's a history of accretion. It's an evolutionary accretion process of the parts as they're coming in time. And we're going to see that when you map where the biospecific fold structures, fold superfamily structures appearing in this time, you have an enrichment, very, very ancient, and an enrichment very, very recent. The red coming up in that mapping. You can do this only because these trees, the trees of parts, have a very peculiar characteristic that is fundamental for phylogenetic analysis. The trees are incredibly unbiased. They are not balanced. Trees of life are usually balanced. They're not completely balanced. They're a little bit unbalanced, and that's very important. We don't understand why unbalance occurs in trees of life. But in trees of parts, they're incredibly unbalanced. And when you have that kind of thing, you have a cone-like tree. And a cone-like tree allows you to turn the tree into a timeline. And this is exactly what we do. With limitations, but we can do that regularly. And that is what we can then assign a fold age to this in billions of years, thanks to that property. So we've used this accretion to study subsystems in general. So for instance, we've concentrated on the ribosome and trace ages of ribosomal proteins, ages of helical systems, because we can do that with RNA too, and produce a model derived from data of the evolution of the ribosome. We've done the same thing with the origin and evolution of aminosulfoname synthetase functions, which are fundamental to describe the origin and evolution of the genetic code. And of course, we were very much interested in patterns of change that will occur at protein level. So we studied domain gains and losses in protein evolution through the time. And all this is thanks to the accretion process ultimately. And we can do this, we can focus on the ATPase motor that I was referring to earlier, and put forth a model of how that motor was generated. It started with a ring structure first, then it added a shaft, then it added a stator, the greenish part of the structure, and they're colored according to age. So the more bluish and green you go, the more recent it is. And so this is an evolutionary model of a molecular makeup of a complex that is made, of course, of domains, and we're looking at this from a domain point of view. So can we use that science strategy, but not look at so much the physical aspect of things? things, but the aspect of sharing, which has been the aspect that we're interested in. So we see Venn diagrams, there are patterns of sharing between organisms. So can we trace patterns of sharing through Venn groups and in the timeline? What do they tell us? Because they carry a lot of interesting information. So what we find is that at the base of that evolutionary timeline, which is close to zero, we have the appearance of things that are universal, as they should. So universal things had to appear at the trunk of the tree of life. So no lineage at that time. And indeed, the ones shared with viruses appear there. But interestingly, the ABE group, which is the group shared only between cellular superkingdoms, appears a little bit. Both of them are most ancient. And of course they unfold all the way till the present because they're continuously producing diversity, all those groups. The interesting thing is that there is a large group of Venn subgroups that are specific to superkingdoms and viruses, and they all appear quite late. They appear halfway in evolution. This coincides with probably the rise of diversified lineages. That's what we are expecting, and that's where the tree of life will fully materialize and will appear there. Interesting thing, the B group that you have there appears before any of the other groups, supporting this idea that bacteria generated diversity unique to bacteria very early in evolution compared to the other superkingdoms. However, the most important thing to consider is that the ABE group that you have there precedes the BE group. And that means that it is quite possible that Archaea was the first cellular superkingdom to start losing structures very early, before even any novelty would have been generated. This is what this is telling us. There's no other explanation for that. Especially because trees are based on abundance, and we can use occurrence to generate this. And very ancient structures are highly abundant, so they're enriched in that whole region. So to lose a structure is a major event in evolution. Archaea have lost many. And so this is basically the essence of what we are finding from that, which is a tree of parts. No problems of character independence here, hopefully, but yet we get this information. Very early emergence of the biosphere, then in worlds that were only structural and communal, taking the ideas of Carl Walls perhaps, during that time, we had only stem lineages. These are trunks that were highly enriched in what we studied, which are four superfamilies, and they were basically losing stuff when they were diversified. We would not generate novelties in whatever entities were being formed at that time. So as you can see, there's a very special pattern there that I'll discuss in a minute. When we look carefully at how the diversity distributes in viruses, in archaea, in bacteria, in eukarya, using this F-index, which is the distribution of structures within the organisms of each of those groups, we see that viruses, of course, are lost a lot. Look how flat that is. So that is indication of reductive evolution to the maximum. And archaea follows with that. A lot of reductive evolution very early in life. Look how that falls immediately down to really low levels. That means very old structures being shared by very few groups of organisms within archaea. Then bacteria and then eukarya. This is just a more detailed view of the reductive tendencies in viruses, showing that the virus-specific folk superfamilies, the green one there, in the third group there, appears clustered exactly at the time where organisms are unfolding all the superkingdom-specific diversity. At that time, we have virus-specific folk families appears in the time. And that is expected, because you have to have a host. And the host has to be in the form of a lineage that is diversified so that the host interaction becomes specific. isn't it? So that's where you have to unfold capsids and all the entities of warfare that viruses use in order to interact with these cells. So they appear all consistently together in that time. And then we can ask the question that I posed earlier. So what about the age of the threads? Now we can add ages to the thread, and if they're reddish, they're very old. If they're bluish, they're very recent. As you can see, there's incredible bias towards older ages. They're less than 0.3 of this age of protoporphyrin. So that means that the threads are very ancient, and phylogenetically we are proving that that is the case through the tree of fold structures. So this is again revisiting what the tree of parts is telling us. And it's telling us that we had first probably a rise of grades, as they are called. These are not really lineages. They have the phenotypes of the trunk lineage, a stem line of descent. And it's only when we have a rise of clades that we start seeing the diversity of our tree of life, and that occurs much later, after we've already uncovered the vast majority of the protein world from a structural point of view. And at that time, we had already possible viral lineages unfolding, or viral grades unfolding at that time. So this is our hypothesis. So if this would be the case, then what happens when we flip the matrix? We use the same thing that we've been using, we just flip the matrix, and then we now ask the question, can we build a truly universal tree of life that incorporates viruses? And will we have this kind of output from the tree? Our trees are rooted. That means that we can provide that directionality of time. And this is what we got, and we published very recently, where we have all the viral lineages coming from the base of the tree of life. And there are 4 domains, and I wrote domains here, but we could call them supergroups, because we should be caution. We should accept caution for mentioning things about classification in general. But they're very distinct 4 groups. The first 2. Consistently in oral analysis have produced paraphyletic patterns. And what is a paraphyletic pattern? It's a pattern comb-like. So archaea and bacteria, both of them, are producing comb-like appearances that unify perhaps some groups of archaea, together, like some groups, large groups of And the same occurs with viruses. We have some groups that are clustered together, like the groups of the Liga Membranales and the Liga Membranales, exception is accepted. And there are close relationships that are well established through sequence analysis, meaning that we are seeing patterns that are consistent with what sequences are telling us about relationship of viruses. But the interesting thing is that at the base of the tree, we have RNA viruses, which was what our Ariadne threads were suggesting, isn't it? So, and interestingly, those viruses have a strategy of segmenting RNA. So that means that, that it is possible that perhaps early genomes were segmented genomes, as Carl Woese anticipated at some point. This is an evolutionary network supports this, and this is again what I was referring to, the evo-PCO analysis, this numerical approach, again showing that same pattern. And instead of looking at it in 3 dimensions, we can use clustering analysis, and again at the base of that tree there you have there, which is a numerical analysis of that space, again shows the routing numerical. So out of 3 main lines of thought, we have basically the virus verse hypothesis, the reduction hypothesis, and escape hypothesis. I would suggest that this data is only compatible with the reduction hypothesis, and so we are basically falsifying the other 2 versions with this data. And this brings a problem. Now we have to define an equivalent less universal common ancestor has to be the ancestor of viruses and cells. And then we need to have a less universal cellular ancestor that unifies all cells together. So now we have to think of how to work on those aspects of evolutionary equation. And this is evidence from geology. So if you go back to 2.9 to 3.5 billion years, that's the time where all this is materializing. And you look at the sediments, and you look at these carbonaceous materials, these are structures that have been explored, taken out of rock, isn't it? And there are two sides of structures. And this is kind of curious. Why two sides of structures? So I just leave this there for you to consider. I've been published in Nature and other journals, and different groups have found this reliable. These are, first of all, we don't know the biogenicity of this, if this is true or not, but our results support this. Two structures of different size will imply a group of cells going productive. And of course, then I missed this. These are kind of the main take-home message of what I put forward. Early cellular origin of viruses, late rise of viral novelties, and ultimately the idea that we have to rethink concepts, general concepts about viruses. And this is important because, you know, especially there's incredible viral diversity in the oceans, never around us, and we are mining. So, what, are they really only pathogenic? Are we having something else? And this is just my acknowledgements, specifically to Rashan Nasir, which have done a lot of work here, and then Phil Kim in Korea has helped us, and we have collaborators that are listed there. And thank you so much for your attention.